Step 2 of the 5 steps of infection is adhesion. So adhesion means that you're going to stick to your host. So it's not enough to just come in, you have to be able to stick. As a matter of fact, you breathe in something like 20 million viruses and bacteria in every breath you take. But for the most part, you're going to breathe all those right back out. Only those that stick have the potential of becoming harmful because they have actually adhered to the surface of the host. There are 2 primary ways that pathogens are going to adhere. One of them is what is called adhesins. Notice, adhesion is a step. Adhesin is different ways you can stick. And these are all in table 10.4. So, if you look in table 10.4, you can see that there are some cellular structures that will help us to adhere, that are considered adhesins. We've talked about some of these. Those fimbriae that we talked about when we talked about cellular structures. Some pili, this is their function. Capsules. Remember, if you are a slimy glycocalyx, basically you're snot. And if you've ever flicked a booger onto the wall, you notice that it sticks to the wall because carbohydrates are actually really good at sticking to things. That's why we make glue out of carbohydrates. And then embedded in that plasma membrane, we saw lots of glycoproteins. So we have proteins that are either peripheral or integral, and sticking out of them are some carbohydrates attached to them. These are called binding factors. So another type of adhesin is a binding factor. And on the host, they're going to stick to lots of different things. So there are sugary molecules on your surface in which these can stick to. Sialic acid is a carbohydrate that a bacteria can stick to on your cells. Heparin. And then we also have proteins that some of these glycoprotein binding factors will stick to. And these are called fibronectin. If you remember, fibronectin, these are some of those proteins that are extracellular to your cells and actually weave into and out of each other in order to hold your cells together. This is what allows the cells to connect in order to make tissues. So there are 3 different types of molecules that the glycoproteins on the bacteria can adhere to on your host cell. A second way that we can adhere as a pathogen is that we can get embedded and become part of that biofilm. So biofilms are collections of bacteria, collections, even more generally, of microbes. They're communities that are part of your normal microbiota. So, if you take up residency inside of our microbiome, then by default you have also taken up and have adhered to your host. It turns out that these communities are actually very well regulated between themselves. This is a term called quorum sensing. Different parts of the biofilm, different microbes within it, will excrete certain chemicals that will then affect other microbes in that microbiome. And depending on what kind of organism gets embedded in that microbiome, these quorum sensing chemicals can actually affect them. One of the examples that they give is the common yeast molecule. So if you can get a infectious yeast in you and within the microbiome of your genitals, the yeast is kept in check. But if it happens to move to the mouth and get embedded in a different biofilm of the microbiome, it can actually turn out to be a lot more harmful and a lot more dangerous. That's all due to the quorum sensing affecting this. And this is another place where if you have something that is typically innocuous but now gets into a different community and gets different signals, it can become an opportunistic pathogen.